Hello students, welcome to readmedprep Academy channel. Today in Classical Genetics Part 3, we are going to talk about Monohybrid Cross, Test Cross, Back Cross, Dihybrid Cross, Dihybrid Test Cross and the gene interactions. Monohybrid inheritance is the inheritance of a single character, for example the plant height. It involves the inheritance of two alleles, alternate forms of a gene, and the alleles belong to a single gene. What is the molecular explanation of Mendel's gene for plant height in peas? The plant height is controlled by a single gene with two alleles. The tall is the dominant trait and the dwarf is the recessive trait. The following facts give a reason for the plant height. The cells of the pea plant have the ability to convert a precursor molecule of gibberellins into an active form GA1. Tall pea plants have one allele, capital L and small e, that codes for a protein. This protein is a functional enzyme which functions normally in the gibberellin synthesis pathway and catalyzes the formation of gibberellins GA1. The allele is dominant even if it is 2Le or single, capital L small e and small l small e. It produces gibberellins and the pea plants are tall. Dwarf pea plants have two recessive alleles, small l small e, small l small e, which code for non-functional protein, hence the plants are dwarf. The dominant allele in the double LE, LE, the L is capital, or a single capital L small e produces tall plants. In the image you can see the tall pea plants with the alleles capital L small e, capital L small e, or capital L small e, small l small e. And the gibberellin, which is a precursor molecule, forms GA1, which is an active gibberellin which helps in the tallness or the increase in the height of the pea plants. The two recessive alleles, small l and e, small l and small e, produces dwarf plants. The dwarf pea plants with the alleles small l small e, small l small e, the precursor is the gibberellin. But this gibberellin does not undergo metabolism and does not form the active gibberellin. Hence the plants are dwarf. So capital L small e allele codes for the functional enzyme GA1, whereas the small l small e allele codes for the non-functional enzyme. Now let us discuss about the monohybrid cross. What is this F1 generation or the first filial generation? It is the second stage of Mendel's experiment. When the F1 generation was Mendel noticed that 787 plants of 1064 F2 plants were tall, while 277 of the 1064 plants were dwarf. The dwarf trait disappeared in the F1 generation only to reappear in the F2 generation. What is F2 generation or the second filial generation? The third stage of the Mendel's experiment. The generation obtained after crossing F1 generation is the F2 generation. What is a genotype? The term genotype is the genetic constitution of an individual or the types of alleles in a single individual. The term phenotype refers to the physical observable character that is present in an organism, the physical expression of an individual gene. Here you can see the differences between the phenotype and the genotype in a pictorial manner, where the flower color or the size of the wings or the height of the plants or the color of the pig. So the phenotype is the physical expression of the character, whereas genotype is the genes that code for that specific character. Now in a genetic cross, the genotypes and phenotypes of the offspring resulting from combining gametes during fertilization can be easily understood with the help of a diagram. That diagram is called Punnett Square, named after a British geneticist Reginald C. Punnett. A Punnett Square is a tool used to predict the percentage of genotypes for the offspring of two known parents. Each square represents a fraction of percentage. Here you can see the image on the right side, where on one side on the top is the parent genes, on the left side of the image is the parent genes, and each square represents 25% of the offspring. So there are 4 offsprings in the boxes, so each box is about 25%. A sort of cross multiplication matrix is the Punnett Square. It is used in the prediction of the outcome of a genetic cross, in which male and the female gametes and their frequencies are arranged along the edges. So here you can see in this Punnett Square, the dominant character is represented by capital B and the recessive character is represented by small b on the one side. And on the top, the dominant character is represented by capital B and the recessive character is represented by small b. So on multiplying each character, you can see capital B will produce capital B progeny. Capital B with small b will produce capital B small b, which is called heterozygous. Capital B small b will produce capital B small b. Small b will produce small b, which is homozygous. So capital B is called homozygous dominant. Capital B small b is called heterozygous dominant. And small b is called homozygous recessive. It is the graphical representation to calculate the probability of all the offsprings and the genotypes in a genetic cross. The law of dominance and the law of segregation give suitable explanation to the Mendel's monohybrid cross. Here you can see the monohybrid cross, where the parent generation is taken as a true breeding line, which is the dominant character is tall, represented by capital T, and the dwarf character is taken as small t, which is a recessive character. By crossing these two plants, the F1 generation is got, and it is represented by the Punnett Square or the checkerboard, where all the 4 plants that are obtained are heterozygous dominant with the genotype capital T and small t. In the F2 generation, when the heterozygous plants are crossed, you get 3 tall plants and 1 dwarf plant. Out of the 3 tall plants, 1 is homozygous, 2 is heterozygous, and the homozygous is represented as capital T, the heterozygous is represented as capital T, small t, and the homozygous dwarf plant is represented by small t. So the offspring in the F2 genotypes is capital T, which is 1 in genotypic ratio, and capital T, small t, the genotypic ratio is 2, and the small t, which is a dwarf plant, the ratio is 1. So 1:2:1 is obtained. So 1 is homozygous tall, 2 is heterozygous tall. So the ratio, the phenotypic ratio that is obtained is 3 tall plants and 1 dwarf plant. Now what is reciprocal cross? In one experiment, the tall pea plants were pollinated with the pollens from true breeding dwarf plants. The result was all tall plants. When the parental types were reversed, the pollen from a tall plant was used to pollinate a dwarf plant, which gave only tall plants. So the result was the same, all were tall plants. So tall, which is considered as a female and is crossed with the dwarf, which is considered as a male, and the tall, which is considered as a female and is crossed with the dwarf, which is considered as a male, and when the mating is done, this is called a reciprocal cross. And the results of the reciprocal cross are the same. Here you can see the reciprocal cross between a green versus yellow pods. On the left side, the dominant character green G, and the recessive character small g. The dominant character is taken as the male and the recessive character is taken as the female. By artificial cross fertilization, we can get the phenotype, which is green, and the genotype is capital G and small g. When the yellow is taken as the male and the green is taken as the female, the yellow represented by small g and the green represented by capital G, on artificial fertilization, the F1 generation will show all plants having green, and the genotype is capital G and small g. So the reciprocal crosses between pure breeding parents produce identical results. So it was concluded that the trait is not sex dependent. The results of Mendel's monohybrid crosses were not sex dependent. The gene for a plant height has two alleles. Tall is represented by capital T and the dwarf is represented by small t. The phenotype and the genotypic analysis of the crosses has been shown by a checkerboard method or by fork line method. So Mendel chose two contrasting traits for each character in his analytical and empirical approach. So it seemed logical that two distinct factors exist. In F1, the recessive traits and its factors do not appear. They are hidden or masked, only to reappear in 1/4 of the F2 generation. He concluded that tall and dwarf alleles of F1 segregate randomly into gametes. You can see in the image on the right side, the dominant character TT, capital T, tall, and a dwarf character small t on the right side of the image. In the F1 generation, there are heterozygous plants formed which are dominant, represented by the genotype capital T and small t. And the gametes that are formed are two types, capital T and small t. In the F2 generation, when the fertilization occurs, you get 3 dominant plants and 1 recessive plant. The dominant plant, 1 is homozygous, 2 is heterozygous. So the Mendel got 3:1 ratio in F2 between the dominant and the recessive trait. He was the first scientist to use this type of quantitative analysis in a biological experiment. In this checkerboard, you can see in the F2 generation, the ratio formed is 3:1. That means 1 is homozygous, 2 is heterozygous of dominant, and 1 is homozygous dwarf. So 1:2:1 is the genotypic ratio, and the phenotypic ratio is 3:1. Mendel's data is concerned with the proportions of offspring. Mendel's analytical approach is truly an outstanding scientific achievement. His meticulous work and precisely executing breeding experiments proposed that discrete particulate units of heredity are present and they are transmitted from one generation to the other. Now these units are called genes. The particulate hypothesis is the idea that parents pass on discrete heritable units. These units initially were called as factors, now they are called as genes. Mendel documented a particulate mechanism through his experiments with the garden peas. Mendel's experiments were well planned to determine the relationship which govern hereditary traits. This rationale is called empirical approach. Laws that were arrived from an empirical approach is known as empirical laws. Mendel did not have a hypothesis that he was testing, hypothetico-deductive method. Instead, his experiments were designed to determine the quantitative relationships from which laws could be discovered. Now, let us move on to test cross. Test cross is the crossing an individual of unknown genotype with a homozygous recessive. In Mendel's monohybrid cross, all the plants are tall in F1 generation. In monohybrid cross, F2 tall and dwarf plants were in the ratio of 3:1. 1 homozygous capital T, 2 heterozygous capital T small t, and 1 homozygous small t. Mendel self-pollinated the dwarf F2 and got dwarf plants in F3 and F4 generations. So he concluded that the genotype dwarf was homozygous, small t. The genotypes of tall plants, capital T and capital T small t from F1 and F2 generation cannot be predicted. To determine the genotype of a tall plant, Mendel crossed the plants from F2 with the homozygous recessive dwarf plant. This procedure was called as a test cross. The progenies of the test cross can be easily analyzed to predict the genotype of the plant or test organism. Thus, in a typical cross, an organism, pea plants, showing dominant phenotype, whose genotype is to be determined, is crossed with the recessive parent instead of the self-crossing. The test cross is used to identify whether an individual is homozygous or heterozygous for the dominant character. Here you can see a heterozygous tall test cross, where from the F1 generation heterozygous tall is taken, capital T small t, and crossed with homozygous dwarf. The capital T small t will produce gametes capital T and small t, and the homozygous dwarf will produce gametes small t alone. And when they are crossed, the genotypes in the F1 generation, capital T small t and the small t is obtained, and the genotypic ratio obtained was 1:1, and the phenotypes were tall 1 and dwarf 1. The phenotypic ratio 1:1 was obtained. By a checkerboard, you can see that when the crossing is done, the 50% plants were tall and heterozygous, and 50% plants were homozygous dwarf. Heterozygous tall is represented by the genotype capital T and small t, and the homozygous dwarf is represented by the small t. Now let us go to another test cross, where a homozygous tall is taken and is crossed with a homozygous dwarf. Generation obtained is capital T and small t, and the homozygous tall is crossed with the heterozygous dwarf. When the homozygous tall is crossed with the heterozygous dwarf, again all the plants that are obtained are completely tall plants, and there are no homozygous recessive plants at all. So these plants are having a genotype of capital T and small t, and they are heterozygous tall plants with the genotype capital T and small t. Now what is back cross? Back cross is a cross of F1 hybrid with any one of the parental genotypes. The back cross is of two types: dominant back cross and recessive back cross. It involves the cross between the F1 offspring with either of the two parents. When the F1 offspring are crossed with the dominant parents, all the F2 develop dominant character, and no recessive individuals are obtained in the progeny. Here you can see in the dominant back cross, where the pure tall is crossed with pure dwarf, and the F1 generation produces all tall plants. And when these all tall plants are crossed with the parent with the dominant tall homozygous, then the produce obtained is all plants are tall. 50% of them are homozygous and 50% of them are heterozygous. If the F1 hybrid is crossed with the recessive parent, individuals of both the phenotypes appear in the equal proportion, and this cross is specified as test cross. The recessive back cross helps to identify the heterozygosity of the hybrid. Here you can see the F1 hybrid capital T and small t crossed with the parent recessive homozygous plant, which is dwarf small t. The gametes obtained by the F1 hybrid capital T small t is capital T and small t, whereas the gametes obtained by the recessive plant is small t. When they are crossed, 50% of the plants are heterozygous tall and 50% of the plants are homozygous dwarf. Now what is dihybrid cross? It is a genetic cross which involves individuals differing in two characters. Dihybrid inheritance is the inheritance of two separate genes, each with two alleles. When two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent to the other pair of characters. Here you can see a true breeding plant with round and yellow cotyledons, and another plant which is homozygous with wrinkled seeds and green cotyledons. The plants obtained in the F1 generation all are round and yellow cotyledons. Their genetic constitution is capital R, small r, capital Y and small y. Genes that are located in different chromosomes assort independently during meiosis. Many possible combinations of characters can occur in the gametes. Now let us discuss about the law of independent assortment in the dihybrid cross, where there is segregation of the gametes. The homozygous characteristic features of round seeds and yellow cotyledon, which is the dominant character, and the homozygous recessive characters, wrinkled seeds and green cotyledon. The parents have the genotype capital R, capital Y. Whereas in the homozygous recessive characters, they have small r, small y. And when they undergo meiosis, the dominant characters produce the gametes capital R, capital Y, and the recessive characters give a gamete RY. When they fuse together by fertilization, they form in the F1 generation heterozygous character. Capital R, capital Y, small y, which have the dominant characters capital R and capital Y. Hence, the seeds are round and they have yellow cotyledons. When these plant produce gametes like capital R, capital Y, capital R, small r, capital Y, small r, small y. Self genes are present on separate chromosomes and random assortment takes place. So 4 different types of gametes in equal proportions are formed by the law of independent assortment. Independent assortment leads to genetic diversity. If an individual produces genetically dissimilar gametes, it is a consequence of independent assortment. Through independent assortment, the maternal and the paternal members of all pairs were distributed to the gametes. So all possible chromosomal combinations were produced, leading to genetic variation. In sexually reproducing plants, organisms, due to independent assortment, genetic variation takes place, which is important in the process of evolution. The law of segregation is concerned with the alleles of one gene, but the law of independent assortment deals with the relationship between many genes. The crossing of 2 plants differing in 2 pairs of contrasting traits is called dihybrid cross. In dihybrid cross, 2 characters, the color and the shape, are considered at a time. Mendel considered the seed shape like the round and the wrinkled, and the cotyledon color like the yellow and the green color as 2 characters. In seed shape, round capital R is dominant over wrinkled small r. In cotyledon color, yellow capital Y is dominant over green small y. Hence, the pure breeding round yellow parent is represented by the genotype capital R, capital Y. And the pure breeding green wrinkled parent is represented by the genotype small r, small y. During gamete formation, the paired genes of a character are sought out independently of the other pair. Here you can see the parents round yellow and wrinkled green on the right side, and the round yellow will give gametes capital R, capital Y, and the wrinkled green will give the gamete small r, small y. In the F1 generation, all the plants will have round seeds with yellow cotyledon, genotype being capital R, capital Y, small y. During the F1 with F1 fertilization, each zygote with an equal probability receives one of the 4 combinations from each plant. The resultant gametes thus will be genetically different, and they are of the following 4 types. Yellow round capital Y R, which occupies 9/16. Yellow wrinkled capital Y small r 3/16. Green round small y capital R 3/16. Green wrinkled small y small r 1/16. So here you can see the gametes that are formed by the F1 generation plant, which is heterozygous, which are of 4 types. These 4 types of gametes of F1 dihybrids unite randomly in the process of fertilization and produce 16 types of individuals in the F2 in the ratio of 9:3:3:1. Mendel's 9:3:3:1 dihybrid ratio is an ideal ratio based on the probability including segregation, independent assortment and random fertilization. Here in this image you can see on the left side the parent, which is round and yellow cotyledon, is taken as the female, and the parent wrinkled seed and green cotyledon is taken as the male, and the genotype is capital R, capital Y, and on the parent too the genotype is small r, small y. The haploid gametes that are produced by the dominant characters are capital R and capital Y, whereas the recessive characters are small r, small y. In the F1 generation, the parent formed is heterozygous. Capital R, capital Y, small y. And in the F1 generation, all the phenotypes are round seeds with yellow cotyledon. And the genotypes, capital R, capital Y, small y. And this parent will produce 4 types of gametes. And when the 4 types of gametes are put on the above and the gametes on the left side of the checker box, we can come to an arrival that 16 plants are formed with 16 different types of characters, out of which 9:3:1 phenotypic ratio is obtained. For example, capital RY and capital RY will produce capital RR and capital YY. Similarly, capital R, capital Y with capital R and small y will produce capital RR, capital Y, small y. Capital R, capital Y with small r and capital Y will produce capital R, small r, capital Y, capital Y. Capital R, capital Y along with small r and small y will produce capital R, small r, capital Y, small y. So like that, if you keep on calculating, we get 16 types of plants, out of which the ratio is 9:3:3:1. 9 plants are round and yellow, 3 plants are round and green. In sexually reproducing organisms, plants from the garden piece to human beings, Mendel's findings laid the foundation for understanding the inheritance and revolutionized the field of biology. The Dihybrid Cross and its result led Mendel to propose a second set of generalizations that we called Mendel's law of independent assortment. Now, something about the dihybrid test cross. The F1 hybrid plant, round yellow piece, is crossed with homozygous double recessive genotype. Wrinkled green piece, small r, capital Y, capital Y. This is called dihybrid test cross, which the ratio obtained is 1:1:1:1. Here you can see the heterozygous parents, the round piece with yellow cotyledon, and the homozygous wrinkled piece with the green cotyledon. When they are crossed, they produce gametes. And the genotypes formed are capital R, capital Y, small y, round and yellow. Capital R, small y, round and green. Small r, capital Y, small y, wrinkled and yellow. Small r, small y, wrinkled and green. The ratio is 1:1:1:1. Round yellow, round green, wrinkled yellow, wrinkled green are the phenotypes. So the phenotypic ratio is 25%, 25%, 25% and 25%. Now, how does the wrinkled gene make Mendel's piece wrinkled? This is explained by the molecular genetics. The protein called starch branching enzyme SBEI is encoded by wild type allele of the gene capital R, which is dominant. When the seed matures, this enzyme SBEI catalyzes the formation of highly branched starch molecules. The normal gene capital R has become interrupted by the insertion of the extra piece of the DNA 0.8 kb into the gene, resulting in small r allele. In the homozygous mutant form of the gene small r, which is recessive, the activity of the enzymes SBEI is lost, resulting in wrinkled piece. The wrinkled seed accumulates more sucrose and high water content. Hence, the osmotic pressure inside the seed rises. As a result, the seed absorbs more water, and when it matures, it loses water as it dries. So it becomes wrinkled at maturation. When the seed has at least one copy of the normal dominant gene, heterozygous, the dominant allele helps to synthesize the starch, amylopectin, an insoluble carbohydrate with the osmotic balance, which minimizes the loss of water, resulting in smooth, structured, round seed. Here you can see in this image the molecular explanation of round and wrinkled piece. The wrinkled gene makes the Mendel's piece wrinkled. On the left side you can see the round piece. On the right side you can see the wrinkled piece. So the dominant allele RR, what it does, it converts the linear unbranched starch amylose into a branched starch amylopectin because of SBEI and forms round piece. Whereas the recessive allele small r does not produce the enzyme and the enzyme is inactive. Hence, it does not convert the linear branched starch amylose into linear unbranched starch amylose is formed. The linear unbranched starch amylose is not converted. So, it remains as it is and it forms only linear unbranched starch amylose. Hence, the absorption of water occurs and it dries as time passes off and forms wrinkled piece. What about the post-Mendelian genetics? Apart from monohybrid, dihybrid and trihybrid crosses, there are exceptions to Mendelian principles, that is the occurrence of different phenotypic ratios. The more complex patterns of inheritance are the extensions of Mendelian genetics. There are examples where phenotype of an organism is the result of the interactions among the genes. A single phenotype is controlled by more than one set of genes, each of which has two or more alleles. This phenomenon is called gene interaction. Many characteristics of the organism, including structural and chemical, which constitute the phenotype, are the result of the interaction between two or more genes. Mendelian experiments prove that a single gene controls one character. But in the post-Mendelian findings, various exceptions have been noticed in which different types of interactions are possible between the genes. This gene interaction concept was introduced by W. Bateson. This concept is otherwise known as factor hypothesis or Bateson's factor hypothesis. The gene interactions can be classified as intragenic gene interactions or intra-allelic or allelic interactions, and intergenic gene interactions or interallelic or non-allelic interactions. Now, what is the classification of gene interactions? First is intralocus interactions, which are called allelic interactions. Allelic interactions are of three types: dominant relationship, lethal genes, and multiple alleles. The dominant relationship may be because of complete dominance, example tall and dwarf pea plants, incomplete dominance, and codominance or overdominance. Lethal genes are of different types like the dominant lethals, recessive lethals, conditional lethals, sex-linked lethals, and balanced lethals. The interlocus interactions, which are called the non-allelic interactions. They may be epistatic or non-epistatic interactions, and they may be additive or polygenic interactions. So today in Classical Genetics Part 3, we talked about monohybrid cross, test cross, back cross, dihybrid cross, dihybrid test cross, and gene interactions. Thank you. Kindly subscribe, like, share and comment our channel readmedprep Academy. Kindly log on to www.readmedprepacademy.com. Our Facebook ID is readmedprep Academy. Our email is readmedprepacademy@gmail.com. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.